Welcome to our Chapter 47 wrap-up. So for this one, I want to go a little bit more into the development of animals, ultimately how we go from after you're pregnant to developing an actual child and taking care of it. So starting off, you've got in your first trimester, you go from an embryo, which is like a ball of cells that eventually becomes a solid ball of cells called a morula, then it becomes hollow called a blastula, and then from there it starts to like migrate cells and what's called invagination, where it forms like a tube that becomes a digestive tract, and that's called gastrulation. From there, you start to develop characteristics like limb buds and organs to where you get to at about week 8 in humans, at least all the basic features are there. So you have the beginnings of all the organs, you have the beginning of the limbs. So it's not so much like you have a little adult at that point, but at least all the differentiation needed to get to an adult beyond just getting bigger in size, and finishing things up to make them functional, that's going to take place. So it's around there that we say, all right, we've got a fetus. This isn't just an embryo now, this is starting to actually take shape. And then once you get to about week 40, is about what you get with a human pregnancy, you finish gestation, that's the carrying of things, and you get to parturition or birth. So if you look over here, you'll see this sheep is budding off a bloody lamb. This is how cat dog was born. No, this is going to be birth, and so you can see that it's giving birth to the lamb, which is about to land on its head, it appears. But luckily it's not too high, so it should be okay. So this process of actually expelling the young one, the fancy term for that is going to be parturition. Now that's going to be preceded by labor, which will be rhythmic contractions of the uterus that will help push out the baby. You'll also see during this process you'll have dilation of the cervix, which allows for the baby to leave the uterus, because if the cervix is still small, you'll see like 1 centimeter or so is like the norm, the baby won't fit. So that's not going to happen. So in humans, you're about 10 centimeters, means that you're good to go, you can actually fit this thing out. And this works by releasing oxytocin, and oxytocin triggers uterine contractions. And uterine contractions trigger more oxytocin to be released, which triggers more uterine contractions more close together. So this is the idea of, I'm having contractions 4 minutes apart, then 3 minutes apart, then 2 minutes apart. This is a positive feedback system. And then after you have the baby, you'll release prolactin and oxytocin, and that triggers the mammary glands in the breast tissue, at least in humans, to ultimately produce milk. And once again, this will tend to be a more positive feedback system, where the more you nurse, the more you release these hormones, the more milk you produce. If you're not nursing, then it tends to scale back, just like if you're working out, you build muscle, but if you stop working out, you don't keep the muscle. It starts to atrophy. Go look at some ex-football players and see how they tend to go from muscular to rotund. Now, next we've got some terminology. So starting off, you've got a zygote. So the zygote's the first fertilized cell. Within it, it oftentimes has chemicals called cytoplasmic determinants. These are oftentimes imbalanced, so when you undergo cleavage or cell division, each new cell that arises doesn't have exactly the same cytoplasmic determinants. And which ones it has and in what concentration can help cause cellular differentiation. So it can start to give us the basics of what am I going to be as a cell that's part of a multicellular organism. And this leads to morphogenesis, the development of form, like I'm going to have 8 legs or 6 legs, I'm going to be this tall, I'm going to have a head that looks like this, as well as in the internal part, you've got organogenesis, where you're going to say, all right, I'm going to develop a liver here, it's going to be this size, I'm going to have 2 lungs, all that stuff. So these ultimately help us build the organism. And then we know a lot of what we know because we tend to study specific model organisms. So this is like mice, there's a particular nematode worm. In plants, there's a Arabidopsis, a little mustard plant. But we don't try to study everything, that's too difficult. So we tend to try to find stuff that's small, fast-growing, easy to care for. And so we say, all right, fruit flies, you're our man. And so we'll study fruit flies a whole bunch, and we can apply that to other insects, other animals, etc. Now, with differentiation, keep in mind we have genomic equivalence. This means all of our cells in a multicellular organism have the same DNA. Doesn't mean we use it the same, but we still have the same DNA. We'll talk about that this early on when we have cleavage, that's those early cell divisions, those will usually start the differentiation process. We talked about those cytoplasmic determinants that tend to get unequally distributed during cleavage that can start to do some of the very earliest things where maybe this one cell is going to develop to be our skin and our nervous system. So it's not those things yet, but it's been kind of predetermined even early on that it's eventually going to be that. And then induction's kind of like peer pressure, where based upon who's around you, that can help determine what your fate's going to be. So depending on what, like, the signaling and the chemicals other guys are releasing, that helps determine what you decide to do. It's kind of like keeping up with the Jones, I guess. Now, a fate map is kind of cool, where with many organisms, even early on, when they're still just this ball of cells, in this case a hollow ball of cells called a blastula, in many cases, even then, you can go through and you can track what one particular cell is going to become. If it's going to become an eye, or if it's going to become part of a segment, or part of a leg. And so we call these mappings of what you're going to be a fate map. So I'm not saying the cell is the leg right now, but one day it might become the leg. And in many times, because of these cytoplasmic determinants, it's only the zygote in an animal that's actually totipotent. And this means that you're like the ultimate stem cell, where you can be anything if you're totipotent. And oftentimes, even early on, they start to say, I'm still going to be a lot of things, what we call pluripotent. So that one particular cell here might be like, I can be, any of the internal organs, but I'm going to be one of the internal organs. If my ultimate goal was to be muscle, it's not going to happen. And that's ultimately the idea here of why this stuff matters, and how early on we can start to understand how this process works better. Now lastly, with vertebrate limbs, as we try to discuss some of our last terminology pieces, we'll see our limbs need to know several pieces of information. They need to know where the cells are at. So they need to know on this XYZ, this three-dimensional system, where am I? Am I on the edge? Am I on the upper edge? Because based on that information, we can use that to spatially organize the individual. To say, all right, feet need to go here, arms need to go here, head needs to go here, eyes are going to go here. To do that, we have to know where things are at, or else it's not going to work. And so this positional information will help us inform our pattern formation, the actual placement of things, and the Hox genes largely oversee this as well. These are the architect genes, so these ultimately determine how many of things are going to go somewhere. So like this is going to be polydactyly, or probably, I think that's actually polybrachally because it's feet, but ultimately having extra toes. And so if you have a Hox gene screw up, it normally would say, let's develop 5 toes. And if it screws up, it can say, let's go for 6, or let's go for 4. And so these genes, while they don't code for the actual toe itself, they do code for how many and where to put the toes. So these architect genes are very important for evolution, because during this developmental process, if I mess up this one gene, one particular Hox gene, it can dramatically affect the morphology, the look, the appearance, the structure of that organism. It's not like I need 100 mutations to go from 2 wings to 4 wings. One mutation could make the organism say, let's put 2 wings here. And you know what? Let's put 2 more here. Because there's still separate genes coding for how to build a wing. All we're doing is activating those genes that will build a wing in a different spot, in an extra spot. Sometimes this doesn't work out well because the muscles don't attach properly and other things can get in the way, but in some cases it does, and it allows for things to end up being like a millipede, where you seem to have stuttered on the whole, let's put a segment with legs, and you stutter a whole bunch, so you're like, let's put 100 segments with legs. We don't have to have a ton of mutations to get there. You can have one or a few mutations, but they'll tend to be in these Hox genes. They'll tend to be in these architect genes.